So welcome everyone to this month's ReproNim webinar. Following our new theme of trying to include biology and technology together in our presentations, we're joined by one of our colleagues and outside speakers. So I will turn the introduction over to JB Pauline to introduce this month's speaker.
Thank you, Dave. It's a pleasure, really a pleasure to introduce Ross Markello. I've known Ross since I've joined McGill University. He's a PhD candidate in Bratislav Music Lab and has done a superb job in many fronts. And the one he's going to talk about is one of it is very dear to my heart and the ReproNim people, which is the flexibility of biological analysis and the ensuring potential non-reproducibility of results in the literature. So I think it's really at the heart of what ReproNim is doing, and what Ross has been doing has been absolutely really stellar in that front. And I'm so impressed by what you've done, Ross. And also personally, it's just a very, very pleasant, extremely pleasant to know you and interact with you. So it's very, very glad that I could introduce you to this crowd. It's a very friendly crowd, and I'm sure you know some of us, but it's yeah. So thank you a lot for accepting the invite. I know it's also a celebration day for you personally, so it's one of those things. And yeah, so thanks again, and the floor is yours.
Awesome. Well, thank you for having me. Really excited to be here to chat about some work. As you said, I think a lot of the stuff that I'm doing kind of does get at the heart of what, hopefully, what ReproNim is all about, and instilling reproducibility in the work that we're doing in neuroscience. So I appreciate the very kind introduction, JB, and the invitation from both you and Dave and to Julie. So yeah, I'm thrilled to be here to talk to you all today about some recent work that I've done with my colleagues on investigating variability in analyses of brain maps. So when I say that, the first question is kind of like, what does that mean? So to start, neuroimaging and kind of neuroscience, but specifically neuroimaging, has this fascination with maps of the brain. And when I say this, I mean kind of spatial topologies of the brain that represent different types of attributes or features or statistical estimates or things of that nature. Basically, nowadays, you read a paper in any neuroimaging journal, you're going to see brain maps. And they're varied in terms of where they come from, in terms of what they represent. And we can go back to the beginning of neuroimaging to find these sorts of brain maps. But even just looking over the last decade at the literature, there's some that have kind of really stuck out, and that I just want to kind of highlight to show you the range of what can be covered. So, nearly a little over a decade ago, there was this fantastic work done by McGill and colleagues that was published in PNAS, and they looked at evolutionary expansion of the cortex, comparing macaques to humans. So what brain regions tended to be expanded in humans relative to macaques. And at the same time, they looked at developmental expansion. So from infancy to adulthood, what regions of the brain expand over time. And then they did some comparisons between these two. You can kind of squint, and you really don't even have to squint too hard to see that they look pretty similar. There's warm colors that tend to be in the same places between the two. Later that year, a group out of WashU released a few new brain maps. One up here by Vaishnavi et al. Was looking at glycolytic uptake in the brain, what regions of the brain are kind of using the most, or are undergoing the most glycolysis. And then later that year, within a year afterwards, the group also released a map that has kind of carried through this past decade and become quite famous. It's a quote-unquote myelination map. So it's showing regions of the human neocortex that tend to have greater concentrations of myelin, as measured by this T1, T2 ratio that they developed based on some imaging metrics. And you can kind of see, areas of the brain that tend to have higher glycolytic index tend to have lower myelination. And so they, kind of made these natural comparisons therein. Since then, there's an explosion. I can point to basically any publication over the past decade and find a brain map that's been published in one of those publications. So here's just a smattering of ones that have been kind of selected at random, but ones that have kind of consistently been called back on throughout the literature. So what happens is when people are kind of developing these maps, they'll oftentimes look back to things that have been done previously and compare, okay, how does this map compare to this one, and so on and so forth. And these comparisons are kind of done all the time. And so just to kind of highlight how that works, and one of the traditional problems with that, I'm taking some work that was released from the lab just a couple of years ago by a really talented postdoc, Dr. Vasquez-Rodriguez. And what she did is she developed a brain map. Here it's shown, we used a parcellation, so each little dot is a different brain region. And what we did is we looked at the relationship between structure and function in the brain. So can we predict functional connectivity from structural connectivity, and how well can we do that in different brain areas? And so we did this, we ran a bunch of linear regressions for different brain regions and got these R-squared values. The R-squared values representing how well function could be predicted from structure. And it kind of looks nice, you know, it's distinct. It looks kind of like there's these anatomical regions that are kind of grouped together. And we thought that that looks quite close to the principal functional gradient, which was first introduced into the literature by Daniel Margulis and colleagues in 2016, and which we've recreated here. And again, you squint, you can kind of see some of the regions here that are shown in kind of dark blue tends to be a little bit more red. And so we wanted to see how similar these two were. And if we run a correlation, we get a Spearman coefficient of negative 0.48, suggesting that they are quite anti-correlated. Unfortunately, if we were to use a kind of traditional statistical test to derive a p-value from that correlation, we would get a really teeny tiny itty bitty p-value, 4.35 times 10 to the negative 59th. And I would argue that up until just a few years ago, it was actually really common to see these sorts of p-values reported in the neuroimaging literature. I've seen things that are 10 to the negative, 100 something, kind of way beyond the possible kind of precision of a lot of traditional statistical tests. And I'm here to tell you that that's not real. It's just not real. And the reason for that is that the data that we're working with is smooth. Our brain maps are smooth or spatially autocorrelated, right? And it's this feature of the brain maps that's to blame for these teeny tiny p-values that we get when we're comparing different brain maps or features from brains. And the reason for that is that as we increase the spatial autocorrelation or the smoothness of our brain maps, we have fewer true degrees of freedom. So that correlation I just showed you, there were 1,000 points on that scatter plot, but there aren't actually 1,000 independent points. And that p-value was calculated assuming that there were. And so it's drastically inflated. And the issue is that not accounting for this leads to spurious correlations. So we'll say, oh, these things are correlated, R equals negative 0.5 with an incredibly significant p-value. But in actuality, it may not be a significant correlation because the degrees of freedom that we have is so much lower than what we think. So just as a little example to highlight how this works, to kind of get an idea of this phenomenon, I'm showing you here two sets of completely random data. So if you look just on the leftmost column, this is complete Gaussian noise, totally random noise that I've plotted on two different brains. And then what I've done is I've progressively increased the smoothness of the data. So as you move left, this is all the same data. Each row represents the exact same underlying data, just progressively smoothed. And what we do is if we correlate each of these columns, they go from no correlation to an incredibly strong correlation between the brain maps. And again, this underlying mechanism, this underlying data isn't actually correlated in any way, shape, or form, but the smoothness leads to this kind of arbitrary correlation. And that is a real problem. If we're developing brain maps, if we're looking at these things in the literature and we're comparing them back, we want to be sure that the comparisons that we're making, the kind of fundamental attributes of the biology of the brain that we're assessing are real. And so a few years ago, there were some methods that were introduced to account for this spatial aspect of the brain. The brain is a spatially embedded system, so when we're doing these comparisons, we need to account for that in some way. The first method was developed by Aaron Alexander Block and colleagues, and they had been using this method since as early as 2013, but it was kind of formalized in 2018. And what they said is, well, here's what we can do. If we have the cortical structure of the brain that people like to present as these little brain plots, we can project it to a sphere. Because it's all a continuous sheet. And then we can just apply arbitrary rotations to that sphere and then project that back down onto a brain. And this way we can generate a null distribution of brains that have similar properties of spatial autocorrelation. The smoothness isn't changing. We're just changing the orientation. And then we can use those to generate non-parametric p-values rather than using the kind of standard traditional parametric distributions. And in that way, our tests will be a bit more rigorous. This is fantastic. I think the field was really happy to see this method introduced, but it did have some drawbacks, and we'll get into those a little bit later. And so more recently, Josh Burt and colleagues down at Yale introduced this really fantastic method where they generate surrogate data that has similar spatial autocorrelation to empirical maps. So here they're showing you two different empirical maps and then surrogate data that they generated from the method that they produced for that. And it's the same principle. You'd use their method to generate a bunch of these surrogate maps, and you'd generate a non-parametric p-value based on the distribution of those. And so these are fantastic. The issue or the question that we asked was, how well do these models actually control like the family-wise error rates when we're analyzing brain maps? So family-wise error rates here being, do we get inflated p-values when we really shouldn't be? And this is an interesting question, at least we think it's an interesting question, because since those two models that I've shown you were developed, there's kind of been an explosion in the field. There's been quite a few other models that have been proposed to account for this issue of spatial autocorrelation in the comparisons of brain maps. And so to highlight these methods, I'm just going to show a little figure where we've taken a toy data where I've just colored different brain regions based on an anterior posterior axis. And then I'm going to show you an example of a map generated with each of these. There are 10 different null models. So the first two models are what I'm going to call naive models. These do not account for spatial autocorrelation in any way, shape, or form. The parametric distribution is, if you're doing a correlation, it will use a student's t distribution or something of that nature. The non-parametric naive model simply shuffles all the brain regions around with absolutely zero idea or correspondence for space. And so there's these spatial permutation models, these rotations that we're generating, we're projecting to a sphere and rotating around. And since the publication by Aaron Alexander Block, a couple different implementations of this rotation procedure have been developed. And so I'm showing 5 here, and kind of you can visually see, I hope my Zoom overlays are hopefully not obscuring the view for you. You can kind of visually see that there are a few areas that are different. In the Baum and Kornblath methods in particular, you see some missing data. So that's kind of the underlying geometry of the brain. What happens is when the medial wall of the cortex, where there is no cortex due to subcortex and the corpus callosum, gets rotated into the brain, some of these methods have to discard that data. And then you can see some other methods kind of don't quite match or keep the spatial kind of structure and integrity of the data quite as well as others. And then there's this family of models that we refer to as parametrized data models. And what those are is this BERT 2020 method is the one that I just showed you on the last slide. And then there are 2 other methods that were developed, actually the same group. BERT developed a method 2 years earlier that they replaced with their 2020 method. And then there's another method called Moran spectral randomization that's been used. It was taken from geospatial statistics. Actually, a lot of the kind of best statistical work I found in neuroimaging is adopted from geospatial statistics. They have all of the same problems of spatial embedding that we have, but they've just been doing it longer. So Moran spectral randomization is a tool that's taken from that and been applied by a few different groups, but primarily a group out of Montreal, where I'm based, to brain maps. So here's the problem set. We have 10 different models that could be applied to these analyses. are they all giving us the same statistical inferences? If I choose one of these at random, am I going to get the same results? We already kind of know going in that that's not the case for these naive models, but for these other 8 models that seem like they could potentially be doing the same thing, we want to know, are we actually comparing apples to apples? And so before we get into this, I just need to really like lay out how using each of these null models works. So we have a problem where we want to correlate 2 brain maps, X and Y. We do is we generate 1,000 null maps from one of those original brain maps, let's say Y in this case. We correlate X with each null map, so we now have a distribution of 1,000 null correlations. We count the number of those correlations that are greater than or equal to the magnitude of our original correlation, and then we normalize by 1,000 to get a p-value. And so this is, if you've ever done a permutation test, exactly the same procedure, but I just wanted to make sure that we were all on the same page before I go into how we're using these null models in all of our analyses. And so to kind of visually depict that, we have 2 brain maps, X and Y. I've just taken from those kind of simulated maps that I showed you before. And we can say that they're correlated to, negative 0.65. We can use one of our null models to generate different null maps. And you can see here, again, this issue of the medial wall being rotated into our brain space. We're going to have to discard this data when we're generating correlations between these things. We generate, we correlate X with all of these null maps. We generate a distribution of correlations, and then we figure out where our original correlation was and how many of these correlation, this null distribution was greater. We generate a p-value, 0.06. So it's not significant, even though it's an incredibly strong correlation, but it is completely random data. So, that's nice. So we wanted to find a way to start with the basics. We wanted to test null model performance in situations where we knew the ground truth. Are these data correlated? Should they be correlated? Should we be finding significant results? If we dove right into kind of empirical analyses, there is no ground truth in neuroimaging. And so we would have a tough time kind of delineating which models are performing correctly. And so in this case, we can simulate data where we know what we expect and test how the models are doing. And we had questions. Does the spatial autocorrelation or the smoothness of the data impact the performance of the null models? And does parcellating data impact the performance? So if we have vertex level data versus parcellated brain data, which is often a common way to represent and kind of reduce the dimensionality of our data, does that impact how these null models are performing? So to answer that question, I just want to highlight that we're going to be using 2 different multi-scale atlases throughout my results, one based on anatomical delineations and another based on functional delineations. So the functional atlas is the Schaefer atlas, which is quite common. The anatomical atlas is the Commun atlas, which is potentially a little bit less common, but it came out almost 10 years ago. And so we just wanted to kind of have one from each, one anatomical, one functional, and multi-scale, so we could really look to see how resolution of our data is impacting these models. And so what we did is we ran some simulations and we calculated error rates. And again, gonna delineate how we did that. We would simulate 2 brain maps at random. For those 2 brain maps, we would run all 10 null models. So we'd correlate them for one map, generate 1,000 nulls, correlate, generate a p-value. So we'd have a p-value for each different null model. And we would rinse and repeat this 1,000 times. So we ran 1,000 simulations. And again, for each simulation, we're running each of those 10 null models. And then we counted the number of times that p was less than 0.05 for each model. So basically, we're going to set a statistical alpha. This is how many times I'm okay with a false positive, in this case, 5%. And we would normalize the number of times that we got a p-value less than 0.05 for each of these models and yield a false positive, a family-wise error rate. So the reason that this works is because we're simulating our maps completely at random. So we'd expect no correlation because there is no underlying relationship between these 2 maps, but 5% of the time, we're okay with getting a correlation that's marked as significant. And so here we're simulating these maps at different levels of spatial autocorrelation. So here's 2 maps that we would simulate. We'd have a correlation of 0, we'd generate a bunch of nulls, and we'd rinse and repeat this again 1,000 times. So this ends up, we're generating millions upon millions of different brain maps here. And we want to see, how the performance of the null models works. And what we find is that there is quite a lot of error in these null models, unfortunately. As a sanity check, we did look at the correlations between our pairs of random brain maps at different levels of spatial autocorrelation. And what we see here is that as the smoothness of our maps increases, the correlation distribution widens. So we're much more likely to get a very strong correlation between two very smooth brain maps than we are between two maps that are representing just kind of complete unsmooth Gaussian noise. This is a sanity check just to make sure that our simulations were working as we expected and we were generating data with realistic kind of levels of spatial autocorrelation. So now let's look at the errors. What happened? What I'm showing you here on the y-axis is the family-wise error rate, or the probability that we got a p-value less than 0.05 across all of our 1,000 simulations. The x-axis is the spatial autocorrelation of the brain maps we're generating, and each line represents a different null model. And so these purple lines are our spatially naive null models. And what this dashed line here, kind of small, apologies, this dashed line represents 5%. So that's our expected type 1 error rate based on our statistical alpha of p is less than 0.05. So everything should, if it's performing correctly, be at 5%. And we see when there's no spatial autocorrelation in the data, it's perfect. When we're working with completely random noise, everything works as expected. Unfortunately, most of the data that we're working with in neuroimaging isn't completely random noise. It's potentially smooth noise or smooth signal, depending on how optimistic you are about neuroimaging data. As we increase the smoothness of our data, though, these naive null models do terribly. So they quickly reach error levels that are completely unacceptable, like above 75% of the things that they're finding significant are not. The blue lines here represent the parameterized data models, and you can see that they kind of do a little bit of funky things, but they do all rise. And at the highest levels of spatial autocorrelation, they're operating at about 25% error rate on average. And then this red line is a spatial permutation model. So the reason that there's only one spatial permutation model shown here for vertex level data is because the differences in these models is only apparent when we're working with parcellated data. So I'm going to show you that in a second. When you're working with vertex data, all of these models completely identical. So we get one line. And what's notable is that even this model, which is doing a bit better than the others, still has an error rate of about 15%. So it's doing about 5 times worse than what we would want. And so, it's a little bit discouraging to see that all of our models are performing worse than the alpha that we would like them to be. If we look at parcellated data, so shown here for the Kamoun and Schaefer atlases, we see very similar trajectories. The spatially naive null models do terribly, very quickly. The parameterized data models tend to do a little bit worse than the spatial permutation models, but everything has an elevated error rate at these levels of smoothness, data smoothness that we expect in neuroimaging. So really, starting at about 2 here, 2 to 3, that's the range that most neuroimaging data is. And so we're seeing that, maybe at the lower ends, but definitely not at the higher ends where we're not doing quite what we want. And so we wanted to ask what's driving these differences between null models? Why are they performing differently if they're hypothetically trying to do the same thing? And so what we looked at is we said, well, the null maps that are being generated for each of these, the ones that we're using to calculate our non-parametric p-values, do they have similar features to the original empirical maps that we're using? Is the spatial autocorrelation consistent? And so to do that, we measured the spatial autocorrelation of our simulated data. So this is shown here just for the highest level of spatial autocorrelation that we set. We measured how smooth our brain maps that we were generating were. And they range a bit. We used Moran's I here, which is a measure of spatial autocorrelation. But you can see that they're quite autocorrelated. The maximum value here is 1, and you get 1 if you have a uniform field. So there is some some variation, but quite smooth data. And then what we did is for one of these simulated brain maps, we took the 1,000 nulls that were generated, and we calculated the Moran's-I of those nulls. And we looked at the difference between the empirical maps and the null maps. And what we find is that the non-parametric maps, the ones that don't account for space at all, totally different than our empirical maps. The rest are a bit better. You know, they center around 0. The text shown here represents the empirical spatial autocorrelation of the maps that we used to generate these nulls. And what we do is, if we can compare, you know, the distributions here with the performance here, we see kind of a mirror. So these parameterized data models tend to kind of underestimate a bit the spatial autocorrelation of the data, and they perform a little bit worse than the spatial permutation nulls that tend to do a little bit better job at estimating that. And so we thought, you know, maybe this is driving it, but there are still some residual differences. This isn't a complete answer, but it begins to get at an answer to why we're seeing differences in these models that should ideally be performing consistently. So to check in, just what we know so far, null model performance varies across both models and levels of spatial autocorrelation. And the naive nulls are just bad. They're consistently terrible and really should not be used. Spatial permutation models, the red lines, the red ones I showed before, seem to have better performance. And it's possible that the variability in performance we're seeing may be driven by the spatial autocorrelation of the null maps generated by these models, but kind of an open question that we don't totally have a great way to answer. And so a natural reaction to this and to a lot of things is that simulations are nice, but what does it look like in real data? How do these models perform in the wild? As I said before, we wanted to run simulations because it's a way for us to have a known ground truth, but there are kind of downsides to simulations. They're very controlled. And so it's possible that kind of real world data may cause these models to perform a bit differently. And so to assess that, we said, okay, we'll work with empirical data sets and we'll apply these models to those and we'll see how they do. But we want to test different types of analyses. So the type of analysis that we were just testing was what's called a correspondence analysis. And it's asking, are two maps correlated? In the literature, there's a really common other type of analysis called partition specificity, which asks, is a spatial feature of a brain map concentrated within a particular partition of interest, i.e. Resting state brain networks or cytoarchitectonic classes, or if you divide up the brain in some way, is my feature kind of circumscribed by the boundaries of this division? And so what we said is we're going to test two different data sets, and we're going to perform one correspondence analysis with an empirical data set and one partition specificity analysis with an empirical data set, and see how the null models perform. So the first one, testing map correspondence, similar to the simulations we did before, we decided to replicate an analysis from the Alexander Block paper that initially introduced this idea of spatial permutations. And there they used meta-analytic coactivation maps from Neurosynth, and they asked, do different cognitive terms have similar spatial patterns of reported meta-analytic activity? So if I have a meta-analytic map of one term and a meta-analytic map of another, and I want to correlate those, are they really correlated, or is it just due to the fact that we're working with kind of spatially autocorrelated, smooth neuroimaging data derived from meta-analyses? So what I'm going to do is I'm going to walk you through the analysis that we recreated from their paper and then show you how the models do. So the first step is just to get the data. Here, I'm showing you different terms from Neurosynth, which, if you're unfamiliar with, is a fantastic tool for doing meta-analyses of neuroimaging data. And on the x-axis is different brain regions. So here, brain regions are different parcels. As I mentioned, we have this parcellated data. For the empirical analyses, we chose to only use parcellated data rather than vertex level data, as I showed you in the simulations, because, as I mentioned before, the 10 null models are only distinguishable with parcellated data. When we're working with vertex level data, all of those spatial permutation null models collapse, and we really wanted to assess the full range. So I'm just going to be showing you results from parcellated data here. So we have this term by brain region matrix for different meta-analytic activations from Neurosynth. We correlate this and we get a term by term matrix showing how spatially similar these different terms are. We use the different null models, whatever they are, to generate different null maps for each of our terms. And we correlate these maps with our original terms to get an asymmetrical null by original term correlation matrix. We do this 1,000 times. We get 1,000 null correlation matrices. We extract the maximum correlation from each of those matrices, and we generate a null distribution. And we use that to threshold and generate a p-value for our original correlations from this matrix. And so what we did is we used this null distribution to effectively threshold this matrix. Which of these correlations are statistically significant based on the null model of choice? And to kind of derive a summary metric from that, we just counted how many correlations survive statistical correction. And so what we can do is we can plot the number of significant correlations that survive correction as a function of data resolution. So shown on the x-axis here, I said that the parcellations that we're working with are multi-scale. And so we're going to look, as we increase the resolution of our data, how do the null models perform? And as we would actually expect, the naive null models increase with data resolution. And that's artificial, right? We're not actually gaining more degrees of freedom or more information as we increase the resolution of our parcellations. We're just dividing the brain up into smaller and smaller regions. And so we have more data points. And so more correlations are going to be reported as significant. But all of these spatially constrained null models, the 8 that we're really interested in, are kind of condensed here. And so what we can do is we can blow that up here and see that there's, they're a bit different from one another. Interestingly, we don't see a division between the different null model families, right? The blue lines are kind of interspersed with the red lines. And so we're not seeing this separation between them as we did with our simulations, which is interesting. And we can do this for our other atlas as well. And we see completely convergent results, which is that the spatially naive null models are an order of magnitude more liberal than our spatially constrained ones. And within the spatially constrained, there is this separation between model performance, but interestingly, here we assess kind of the spatially constrained null models are quite consistent across data resolutions, which is reassuring for their use. So the next analysis, the final analysis that we wanted to test was testing partition specificity in an empirical data set. And so here we chose to replicate an analysis from Bertha 2018 in published in Nature Neuroscience, Bertha et al. 2018 in Nature Neuroscience, and they used the T1-T2 ratio, or the myelin map I actually showed back on the first slide from the Human Connectome Project. And they asked, is T1-T2 circumscribed by, first, the Yo-Crenin resting state networks? These are kind of canonical intrinsic functional connectivity networks that were derived about a decade ago, and or von Economo cytoarchitectonic classes. So about a century ago, von Economo kind of divided the brain up into the different regions based on their cellular architecture. And so we have kind of large swaths of cortex that are more or less similar to one another. And we wanted to see whether or not those divisions circumscribed this myelin map. So again, same procedure. I'm going to show you kind of the analysis overview and then the results. So here we have our parcellated T1-T2 data from the Human Connectome Project. We have, in this case, I'm showing you the 7 Yo-Crenin resting state networks. And what we do is we calculate for each of these networks the average values of the different parcels in the network. And we can get, these T1-T2 values for the different networks. And then we run our null models and we generate 1,000 null maps for each model. And we can use these, do the same procedure, average within the networks, and we can get null distributions of T1-T2 values for each network. And then what we can do is the same thing. We can ask, are the empirical data significantly different from, or significant compared to these null values? And we can reduce these to a z-score. So we z-score our empirical data by the null distributions to get a kind of single summary statistic for each network. And so when we do that, we get this nice bar plot. We can see regions that are kind of under-enriched versus, or networks that are under-enriched or over-enriched, and networks that are significant, or where, in this case, the null model is saying the T1-T2 value in somatomotor network is significantly over-enriched compared to other networks. So we do this for our naive models, and for the naive null models, shown here just for the Commune Atlas, it says that every single network is either significantly over or under enriched for T1-T2, which we just know is not the case. It's not how this works. You can't have everything over or under enriched. When we run it for our other null models, we get significantly more conservative results. So some of these, each bar here is showing a different network Again, the red ones are significant, and we can see some variability between the null models and what they're marking as significantly over or under enriched. We can repeat for the different parcellation that we have, and we see kind of, again, significantly liberal results for our naive null models, kind of variable results for the rest of our spatial null models, and then interestingly, some differences between the atlases here. And so to kind of tease that out, we decided to take a look at the results for both atlases across all resolutions. I didn't want to do bar plots for that. That would just be, hundreds of bar plots. So we went to a heat map resolution where the y-axis is a different atlas and or resolution, and the x-axis is a different network. And the values in the cells are the z-scores, just the heights of the bar plots from before. And then cells that are kind of highlighted in black outlines are ones that are marked as statistically significant. So we did this for all of our 10 null models for the Euclidean resting state networks. And you can see kind of, these differences between the models are consistent across the atlases and resolutions. We also repeated this for the von Economou-Siderechtectonic classes, shown in this bottom row. And interestingly here, there's actually significantly more agreement between the different null models. And that may just be that T1, T2, which is a microstructural attribute of the brain, is better delineated by the von Economou-Siderechtectonic classes than it is by the Euclidean resting state network. So there's this potential to kind of, use multiple null models to generate some sort of consensus idea about the brain. But so we see this consistent differences between null models. We ran these null models on empirical data sets, and in summary, what we find is this. Naive null models are completely inappropriate for significant testing in neuroimaging. They're still used a bit throughout the field, and they consistently yield incredibly elevated type 1 error rates. So you're going to find things that it says are significantly related, when you're comparing brain maps, that just aren't. And so really their use needs to be actively discouraged in new work. Among spatial null models, the appropriate selection of null model is going to depend a little bit on research context. So what we find is that the spatial permutation null models, the ones that are rotating the brain, tend to be a little bit more statistically accurate, but can only be used with cortical surface data. So if you have subcortex or cerebellum or volumetric data, these do not work. And there's this issue of the medial wall gets rotated into the brain, and you're potentially losing some data. Parameterized data models are more widely applicable. You can use them with vertex or voxel-wise data with subcortex and cerebellar, but they yield slightly increased type 1 error rates. So it's a little bit of, 6 of one, half dozen of the other in terms of you got to pick your poison. You can use spatial permutations, but you're limited to cortex. You can use parameterized data models, but you may be suffering from a little bit of an elevated false positive rate. Reassuringly, we found that spatial nulls were largely parcellation and resolution invariant. So while there were some differences between the parcellation schemes that we used and maybe a little bit across the resolutions of those multi-scale parcellations, by and large, the results are consistent, which is nice and hopefully will help encourage researchers to use these models more willingly with the data that they have. Finally, I want to highlight, and I know this is kind of the point of the ReproNim webinar series, kind of principles of reproducibility that we kind of strove for throughout this work. So first, all of the code for this, all of the analyses for all of the data processing, analyses, and visualization is openly available on GitHub. So here I've just kind of condensed the link, but it's obviously github.com/netneurolab/Markello_spatial nulls. All of the data that we used came from open source repositories. So Neurosynth is an open source repository. HCP, you need to sign a data usage agreement, but it is also open source and very easy to access. And then obviously the simulated data we generated ourselves using the code on GitHub. We ran all of our analyses in a Singularity container that you can find on OSF that we've posted here. We also posted the primary data outputs publicly on OSF and on GitHub as well. And the primary data outputs here, we generated obviously a ton of data. And this is just kind of the most condensed forms of the data, effectively what we used to generate the figures that I showed in this presentation. So the line plots, the bar plots, things like that. we've published all that. The rest we didn't because it was so much, but you can programmatically reproduce it using the code here. And to help you do that, we did something that I've been trying to do in my projects that I've been publishing, which is provide a walkthrough detailing the code usage, which we've hosted as a kind of standalone website on Netlify. It's linked to from the GitHub, so you can kind of easily access it there. And what this does is it just highlights basically working with other people's code can be a real pain in the butt. And so we try and walk you through where our code is, what our code is doing, how you should maybe use the code to reproduce the analyses. For example, for the simulations, that took thousands of compute hours. And so it's impossible to do on a personal, like, PC or a laptop. You need to use something like a high performance compute or a computing cluster. And so we have example scripts for how you would submit those jobs, why you would choose one script over the other for running those on that sort of resource. And so that's available here as what we're calling a walkthrough. Finally, I'd like to acknowledge, my collaborators in lab who supported this, all of our data and funding sources, and then just leave you with this, we're going to have a discussion now, but I'll leave these up as we discuss about why implementation is so important when you're doing analyses, biological kind of real world analyses of neuroimaging data. The implementation of models really has a meaningful impact. And so we need to be considered in how we're doing this. And so we need to kind of occasionally maybe run these sorts of analyses that we ran here to make sure that we're comparing apples to apples rather than apples to oranges. So thanks so much for the time and attention. Looking forward to discussing this stuff with all of you now.
Fabulous. Thanks so much for going through that. I see Jarek has already ventured a question in the chat. I don't know if you can see that or if Jarek wants to speak that question. And then
I'm not sure if it was a question, but it was a comment which I could form into a question.
Sure.
What many don't know is that there is closed form specific equation for distribution of correlation coefficients. And I found it long ago while just trying to match distributions, which I saw using SciPy. I forgot dist match or something like that. So there is nice functionality which would, given a distribution, find me closest matching one among those available, right? And I found that there is distribution for correlation coefficient. And it's quite amazing, right? Because you could just plug in, how many degrees of freedom or points if they're independent, right? Pretty much uncorrelated. And you could see how the distribution forms. And that's why I started to ask question, like, if you have two values, how many possible correlation coefficients could you get? And just to cut it short, 2 minus 1 and 1, right? So when you add third one, you get a little bit of the middle point, right? But the point is that for a while, you get a distribution with really heavy tails.
Yeah.
And that's why I wondered, in your cases, especially when you start smoothing, right? Instead of just looking at, empirically looking at the tails, right? What could have been done is to match what would be this inherent coefficient of independent, size or dimensionality to some degree. It's not dimensionality really, but how many degrees of freedom you have in the data, given your permuted data, and this, distribution function, right? And that would have already given an idea that, apparently, if you have this, it's like you had 3 time points, right?
Yeah.
And then it could have delivered clear message. But then it also introduces a question, if you see that your distribution doesn't match in terms of shape to any of those, right? So you found that the closest one, let's say 12, right?
Yeah.
And then you start to look at the distribution that even after permutation, this permutation rotation doesn't match. It could be because there is indeed data, right? So it's not really random, maybe, or maybe there is additional effects. And that's why I wondered if you try to characterize those distributions in terms of, divergence from some canonical one, or verify that your original one on raw data, or not raw, simulated noise data, right? They kind of would follow this R-dist model, or where would it break and possibly why?
Yeah. So it's a good the functionality in sci-fi always amazes me. And so it is really a really interesting point. I think kind of last bit that you brought up, where it's what happens when your distribution doesn't necessarily match, is something that we've had to think a lot about. And basically the idea is that these models, all of the models we tested, assume that space is stationary. And so that, 2 points that are, 10 millimeters apart on temporal cortex will have the same relationship as 2 points that are 10 millimeters apart on the cingulum. And in reality, that's probably not the case. We're probably dealing in the real world with neuroimaging data with spatial non-stationarities. So the smoothness of our data is varying depending on where we are in the brain. And in that case, we're not going to match the distributions well with these kind of simulations, right? Like our simulations are assuming the way we generated the simulations was using Gaussian random fields that we just added this kind of alpha parameter to that we could raise the spatial autocorrelation of these fields with. And so this idea of spatial non-stationarity and kind of destroying all of these principles that we're basing these null models on is something that we as a field haven't yet grappled with. And so we thought a little bit about it. We in our discussion kind of point to some future directions. How might we begin to deal with this? There are some methods that could work. Work from Michael Breakspear's group looking at kind of wavelet-based decompositions might be a little bit more robust to spatial non-stationarities because they're multi-scale. But yeah, it's definitely an interesting point. And I agree. I think that the looking at the distributions that we generated and comparing them to see kind of how many degrees of freedom are we estimating that we have is definitely a really nice way to kind of show that we don't have as many as we think we do.
We could take bets now. Let's say with that smoothness of 3 where you get to 0.9, my bet would be that you would get just like 4 or 5, maybe.
Yep.
Great, thanks. I encourage others from the audience to bravely unmute yourself or enter a question.
Yeah, I have a question from left field, actually, which is I got distracted by something that you showed, and so my brain went off in a little different direction. And I'll just say I have a background in neuroanatomy that goes back a ways, so it predates my experience of neuroimaging. But when I looked at some of your maps, I started thinking, I wonder, is there somewhere in your data, or is there a way to use some of this methodology to consider the question of whether, this is intuition on my part and it may not be right, but whether functional connectivity maps and anything based on those, which I think of as really statistical maps, may in fact actually be smooth in a very different way from the mapping such as von Economo or any other map that's actually anatomically derived on cellular parameters. So I would expect those to actually I do expect those to look different. And I just wonder whether there are some biologically related questions to what people are looking at and using their maps for that you could inform with some of these methods. That's so interesting to me. I don't know if you have any thoughts on that. I honestly do realize it's coming from left field, but I'm just fascinated by this.
Yeah, no, I mean, I agree. I think that this is an interesting concept to think about and for the field to kind of grapple with. The methods really do ask us a lot to think about the assumptions of our data. So, especially those parameterized models that I was talking about, they're basically like, well, the thing that's important for the data is just the spatial autocorrelation. And that may be true for some types of data, but it may be very untrue for other types. And I think that because these methods are so new, we haven't really begun to use them to interrogate how, we haven't begun to use them to try and inform our biological understandings of the data that we're working with. I do think it's interesting, that you bring up functional connectivity and kind of functional data, because oftentimes when we're using functional data, we smooth it purposefully when we're processing it, versus a lot of the times the data that we're bringing from neuroanatomy, we purposefully don't, because we're interested in this fine-grained information that is so kind of distinct and the boundaries of which are so sharp. And so I think it is an issue of the field just maybe needs to be a little bit more aware of what they're doing. And it's partially a principle of functional connectivity data is derived from, bold, and the bold signal is just so diffuse, right? That like it's a bit of a hammer that we've invested millions upon millions of dollars into measuring. But yeah, I think that there is room for that. I haven't personally thought about it too much. This work, we did just came out. We were really interested in just kind of doing the benchmarking, but I would be really thrilled to see people kind of using these ideas to inform our understanding of the data that we're using and kind of what the biological underpinnings would be.
Yeah, I think it's really interesting. Yeah.
Other questions from the floor? I guess I'll use up the transition time while we're waiting for others. So is there a take-home message of what I should do differently next time I run a amygdala to prefrontal gyrus correlation in my resting state data?
So for resting state data, I mean, if you're just doing seed-based connectivity, it's a bit different, right? But I think a lot of the sorts of correlations that people do can benefit from this, right? So if we think about a functional connectivity matrix, for example, a lot of times you'll see people take a functional connectivity matrix, extract the upper triangle and correlate it with some other matrix. And that still needs to be corrected for with these methods. You'd go back to the beginning, you'd kind of you'd have to shuffle the rows of the matrix using one of these methods. So rather than just doing it randomly, you'd rotate, you'd reassign, and figure out which rows need to get replaced where. But that needs to be done. And so it's kind of one step removed logically. You're not just doing map to map, you're doing this connectivity to connectivity or whatever. But you do need to be doing that, especially there, because when you're doing connectivity matrices, you have thousands upon thousands of points, right? It's n times n minus 1 over 2 is however many different points you have, and they are not independent in any way, shape, or form. So these methods certainly have application when you're doing those sorts of things. Seed-based connectivity, if you're just doing, kind of time series of one region to time series of another, maybe a little bit different, but you do there also need to get into accounting for spatial temporal autocorrelation. Functional data has, that entire other dimension that we weren't considering here. But as I mentioned, I think in response to one of the other questions, these wavelet-based methods that Michael Breakspear's group has come up with kind of try to tackle both spatial and temporal aspects. And so I think they are really promising. We couldn't address them here because they're not well defined for surface level data. And all of our comparisons were using surface data. And those methods are really only developed right now for volumetric data. So we have to find a way to kind of bring them up. Working with surface representations of data, the triangular meshes and things makes the math infinitely more complicated based on my incredibly limited background in math. So we decided to leave that for future heroes to try and tackle in work.
Great. Any more questions from the audience? Well, hearing none,
yeah. I see a question from Chris. Chris, do your findings have any implications on the family-wise correction methods for mass univariate shirts like MDPA, such as cluster correction? I think that those have been developed in parallel to these because they are dealing with something a little bit different. They're trying to tackle similar principles, right? Like we're not actually running, 10,000 independent t-tests when we're doing, a group level, GLM. And so in that case, but because we're doing, we have one sample that we're collapsing across, we're not comparing spatial maps to one another. These don't necessarily apply. I think that it's good that these have developed independently because they fit the problem space of these kind of two different things. It is an interesting question though. I haven't really thought about whether or not one could inform the other beyond just saying, yep, we don't have as many degrees of freedom as we think we do. So yeah, interesting question for the future. Right now, I think my thought would be no, but I'll think about it.
Great. Thank you, Chris, for the electronic question. And before I stomp on anyone else's questions, anyone, last chance, going once, twice, thrice? Okay, well, thank you so much, Ross, for joining us. This is fabulous work and already taking a lot of the reproducibility principles, to heart. And again, it's great for us to see additional ways to solve reproducibility while we work on, ReproNim works on some. It takes a village to do all of this, and it's great that your work is taking advantage of some of those theoretical and philosophical types of approaches.
Yeah,
next month. Oh, thanks. Yeah. Next month we'll be joined by Christina Rapuano, talking the details. I'm not exactly sure, but probably kids and probably development and probably ABCD like data. So stand by for more details as that comes along. Thank you, Chris. Thank you, audience, and we will do it again next month. Stay safe out there and keep reproducing.